So ammonia metabolism we are going to study. These are the various sources that add to the ammonia present in the body, and ammonia, as it is a toxic substance, it has to be neutralized to urea, then it is excreted. So overall, where the ammonia comes from and where the ammonia goes, in what form it is excreted, that we are going to see in this lecture. So sources of ammonia formation, the very first is the bacterial action in the intestine. I hope you can see now the slides changing also. Okay. So bacterial degradation, the bacteria present in the gut, they have the enzyme which hydrolyzes urea, and urea is then converted or produces two molecules, that is, one is ammonia and second is carbon dioxide, and that ammonia is again absorbed into the blood, and that adds to the total ammonia in the body, and once it is absorbed, that ammonia is transported to liver for its further metabolism. The second source of ammonia is trans-deamination. Amino group of most amino acids are funneled to glutamate by means of transamination, followed by oxidative deamination, which rapidly forms free ammonia. As we have seen the transamination reaction, one amino acid is converted to, or one amino acid donates its amino group to a keto acid to form the second amino acid and second keto acid. In this case, this second amino acid, in almost all the many of the transamination reaction, the second amino acid form is glutamate, and that glutamate, by accepting obvious amino group or adding the amino group to alpha ketoglutarate, it is converted to glutamate, and this glutamate then undergo the deamination reaction, in which from that glutamate the amino group is liberated or separated. That adds to free ammonia in the body. As you can see, the first amino acid, okay, and the first keto acid, and by the action of the transaminase, this amino acid donates its amino group to this keto acid, right? So this amino acid and these are the substrate upside, and these two are the products. So this amino acid donates its amino group to this alpha ketoglutarate. So the amino acid itself gets converted to a keto acid, and the alpha ketoglutarate, by accepting the amino group from this amino acid, alpha ketoglutarate is converted to glutamate, and this glutamate is the major amino acid which is formed during transamination reaction of many amino acids, and this reaction, as we all know, it is catalyzed by transaminases. Then next, what is the fate of this glutamate? It undergoes oxidation. The oxidation may be there are two types of oxidation. We'll see, I think details are there, oxidative and non-oxidative deamination of glutamate. So one of the enzyme, oxidative deamination, is caused by the enzyme glutamate dehydrogenase, from which it is hydrolysis of glutamate by addition of water and using the reducing equivalent NAD or NADP plus. Two hydrogens are accepted by this NAD or NADP to form NADPH, and ammonia is liberated, and the remaining compound, as glutamate is formed from alpha ketoglutarate and ammonia. So ammonia is liberated, that means glutamate is regenerated, and this glutamate again utilized for the next set of transamination reaction. So trans-deamination, it is coupled reaction, coupling of transamination and the deamination. So by transamination, the glutamate, a common amino acid glutamate is formed, and from glutamate then ammonia is liberated by oxidative deamination. It is oxidative because the reducing equivalence is generated, that's why oxidative, and alpha ketoglutarate is regenerated. So this is one of the source of ammonia. Then next is deamination, obvious. So we have studied transamination separately. Trans-deamination is combination of trans, or these are connected, always connected, transamination and deamination. So what deamination is? It is removal of ammonia The glutamate is formed in the transamination. So the ammonia or amino group present in this glutamate is liberated in the form of ammonia, and it is coupled with the formation of a reducing equivalent. That's why it is oxidative, because reducing equivalent is generated oxidative. If it is utilized, reductive. Okay, so this is the deamination reaction where ammonia is liberated along with oxidation. That's why oxidative deamination. So in this, the same enzyme catalyzes both the reaction glutamate dehydrogenase. The glutamate in the first reaction or the first step by the action of glutamate dehydrogenase, as you can see, the hydrogen from this carbon and one hydrogen from the amino group are accepted by NAD to form NADPH, and the compound that is formed is called as alpha iminoglutarate. And by further hydrolysis of this alpha iminoglutarate by adding H2O, the amino group present at this position is liberated as ammonia. The oxygen from the water attached here and the two hydrogens are accepted by this amino group to form ammonia. This reaction is again catalyzed by the same enzyme glutamate dehydrogenase. So this is two-step reaction in which glutamate by oxidation is converted to alpha iminoglutarate. By oxidation it is converted to alpha iminoglutarate, and in the second reaction catalyzed by the same enzyme glutamate dehydrogenase, the alpha iminoglutarate is hydrolyzed to alpha ketoglutarate and ammonia is liberated. So it is deamination coupled with oxidation. Second is non-oxidative deamination. It is called non-oxidative. As you can see in this reaction, there is no generation of any reducing equivalent like NAD, NADP, NADH, NADPH or FADH2. No reducing equivalent is generated, so it is non-oxidative. For example, L-cysteine, by the removal of because cysteine is a sulfur containing amino acid. First, the sulfur group is liberated as hydrogen sulfide by the action of enzyme desulfhydrase. So sulfur group is liberated as hydrogen sulfide and it is converted to amino acid. Similar to imino form of glutamate, the cysteine is converted to its amino form, amino acid of cysteine, and from this again by hydrolysis like this same, the ammonia is liberated and the remaining carbon skeleton becomes pyruvate. Like glutamate liberates ammonia and itself gets converted to alpha ketoglutarate. Cysteine, it liberates ammonia and the remaining structure forms pyruvic acid, but the requirement is the removal of the one of the mineral, that is just a minute, someone is not able to join. So L-cysteine is converted to pyruvate by removal of ammonia. Similarly, the amino acid serine, as it is a hydroxyl group containing amino acid, so the enzyme dehydrase removes that extra hydroxyl group in the form of a water molecule. Itself get converted to amino form of serine, the amino acid, and by similar way the ammonia is liberated and the carbon skeleton is converted to pyruvic acid or pyruvate. So these are the two forms, oxidative and non-oxidative deamination of removal of the ammonia. Next source of ammonia is from glutamine. We know the amino acid glutamate. Glutamate it is one of the acidic amino acid. So it has one amino group and two carboxylic groups. But by accepting extra amino group, the glutamine has, when it has two amino groups, it is converted to glutamine. The glutamate ideally has one amino group and two carboxyl groups, but by the addition of one extra amino group produces glutamine, and this glutamine then transported wherever it is required, mainly to the kidneys, and the action of the enzyme glutaminase removes one of the amino group from the glutamine and convert it to glutamate. So the ammonia is liberated. From purines and pyrimidines like adenosine, the enzyme adenosine deaminase acts on the adenosine and removes one of the amino group attached to it, and adenosine is converted to inosine. The guanase, we have seen these reactions, two reactions in the synthesis or the catabolism of purines in the formation of uric acid. So the guanosine by the action of enzyme guanase, it liberates ammonia and converts itself get converted to xanthine. Similar way the pyrimides undergo catabolism by that CPS2 enzyme and further reactions. The end product, unlike the purines, the end product is uric acid, but ammonia is liberated in between the reactions. But the end product of pyrimidine metabolism is ammonia itself along with the carbon dioxide. From amines and other nitrogen containing compounds also, like urea, the other the nitrogen acids and all, from these ammonia also added to the body or to the blood. The ammonia toxicity, because ammonia, as I told you before, is toxic substance. The normal range is very minimal if we compare with the other substances. For example, urea. We are going to study urea. The normal range is 15 to 40 milligram per deciliter or milligram per cent. It is in milligrams. But if you see the normal range of ammonia, it is 10 to 80 microgram per cent or 10 to 47 micromole per liter. It is very minimum. Why? Because ammonia is very toxic to brain, hence rapidly removed from circulation and detoxified by liver and converted to urea. The urea, which is non-toxic substance, which is then excreted by kidneys. If ammonia level exceeds the normal range as specified here, if it exceeds the normal range, it can reach the brain, it can easily cross the blood-brain barrier and shows its toxic effect on brain. It damages the brain cells. That's why the ammonia needs to be converted to non-toxic substance, and that non-toxic substance is urea, and the conversion of ammonia to urea happens via the pathway called as urea cycle or ornithine cycle in the tissue, that is liver, only in the liver. So hyperammonemia, it is called whenever there is elevated levels of serum ammonia than the normal range. It can be acquired or it can be genetic. Acquired, like liver disorders or renal disorders, because the conversion of ammonia to urea happens in liver. So any damage to liver stops this conversion and ammonia is accumulated, leading to hyperammonemia. Second is renal disorders. In renal disorders, the urea is not excreted properly. Some amount of ammonia is also excreted in the kidneys, in the urine, right? That's the characteristic odor to the urine of the humans, ammoniatory. That's why we are ammonioteric. So the renal disorders affect the excretion of urea as well as ammonia. So by backflow, these are accumulated. So it may stop the conversion of ammonia to urea. Ammonia is accumulated, leading to hyperammonemia. The second cause of hyperammonemia is genetic defects of urea cycle enzymes. There are 5 reactions in the urea cycle. Any and each reaction is catalyzed by a specific enzyme. Any defect in any of these 5 enzymes may lead to blockage of the urea cycle, that is the conversion of ammonia to urea. So ammonia accumulates, leading to hyperammonemia. And also the alpha ketoglutarate, this we will see later. So decrease in this reaction shown here, because alpha ketoglutarate, which is a metabolite of TCA cycle actually, whenever there is excess of ammonia, hyperammonemia condition, so alpha ketoglutarate binds to this ammonia and get converted to glutamate. So glutamate is accumulated here, but that may lead to deficiency of alpha ketoglutarate, which is metabolite of the TCA cycle. So as alpha ketoglutarate, if not present as the metabolite of TCA cycle, it will decrease the rate of energy production. So ATP generated is very less in the brain. and this mechanism leads to damage to the brain cells due to deficiency of energy. Symptoms of ammonia intoxication include tremors, slurring of speech, blurring of vision, and convulsion as through the brain damage or brain complications. The complications include encephalopathy leading to coma. Outline of treatment: the low protein diet is prescribed to these patients because ammonia is nothing but the end product of protein metabolism. Proteins are converted to amino acid degrade ammonia then converted to urea. But due to non-conversion of ammonia to urea, ammonia accumulates. So if we stop the protein in the diet, there will be less amount of production of ammonia. The antibiotic neomycin is used sometimes because one of the source of ammonia, if you have seen the first source only we discussed, is the bacterial action of enzyme action on the urea that forms ammonia that adds to blood. So to neutralize these bacteria, neomycin antibiotic drug is used. Then the lactulose, it is a synthetic disaccharide. It is used as a treatment for the constipation. So it acidifies the colon and reduces ammonia in the gut. So biochemical defect of hyperammonemia, in the intestine, ammonia is absorbed from the gut, and ideally it is supposed to transport it to liver for its metabolism, for its conversion to urea and then excretion through kidneys. But it bypasses the liver and it directly goes to brain. Bypass the liver via portosystemic shunting and it goes to brain, and due to this the ammonia absorption or ammonia transport into the brain increases. That leads to conversion of the alpha ketoglutarate from TCA cycle to glutamate. That leads to hampering of the TCA cycle and less ATP production. So less energy, the cells undergo the damage. So as we have seen, alpha ketoglutarate is converted to glutamate. Glutamate again accepts one amino group and it is stored in the form of glutamine. In this condition, this alpha ketoglutarate is deficient. So TCA cycle won't be complete. If the TCA cycle is not complete, ATP production will halt. So increased ammonia production, shift from glutamate to glutamine again. That may lead to cerebral edema and oxidative stress because glutamate has other functions too. It can form one of the neurotransmitter GABA. Okay. So the production of that GABA is stopped. That may lead to various other complications. Transport of ammonia, how ammonia is transported? Ammonia is never transported with blood in the free form because it is toxic. If present in the free form, it can easily cross the blood-brain barrier and damages the brain cells. Instead, the ammonia is transported in the form of glutamine and alanine. Alpha ketoglutarate accepts the glutamate and glutamate again accepts the amino group to form glutamine, and pyruvate accepts the amino group to form alanine. So these two are the major forms of transport of ammonia. Glutamine is a major transport form of ammonia. It enters liver where it releases ammonia by following reaction, by the action of glutaminase. This reaction we already studied. This glutamine once formed, transported to liver. Glutaminase breaks or liberates the ammonia, and the second product formed or liberated is glutamate. The second transporter is alanine. During prolonged exercise and fasting, alanine transported to liver and amino group of it is released and converted to urea. So there is one cycle. So alanine is amino acid. During prolonged exercise and fasting, the alanine present in the muscles, it is converted to pyruvate and ammonia, and that ammonia undergoes urea cycle to form urea, and the pyruvate that is formed, it is through blood transported to liver, and in the liver that pyruvate has various fates. it may undergo formation of glucose by gluconeogenesis, and glucose may undergo glycolysis if energy is required, or that glucose is again transported to muscle. So alanine is converted to glucose, glucose is again converted to alanine. So this cycle is called as glucose alanine cycle. These are the few MCQs. You can discuss it later. The fates of ammonia, what we are seeing is sources of ammonia. Now ammonia is formed from various sources. What is the fate? What happens to that ammonia once it is present in the body or formed in the body? The principal quantitatively important route for the detoxification of ammonia is the formation of the non-toxic substance urea. And that non-toxic substance urea from ammonia is formed in the cycle called urea cycle, or it is also called as ornithine cycle. So overall, to define urea cycle, the precursor or the initial substance, the ammonia, obviously, because we have to detoxify this molecule, the toxic ammonia, and ammonia is one of the substrate along with the carbon dioxide. Okay, so the first reaction happens in the mitochondria, and the site of production of carbon dioxide is the TCA cycle, because acetyl CoA enters the TCA cycle and energy is liberated along with carbon dioxide. So the carbon dioxide from mitochondria generated in the mitochondria along with the ammonia, these two combines to form carbamoyl phosphate by the action of enzyme carbamoyl phosphate synthase 1. In the previous lecture only we have studied about carbamoyl phosphate synthase 2, which is the enzyme of pyrimidine synthesis, right? So this carbamoyl phosphate synthase 1 is responsible for formation of carbamoyl phosphate. Same product is formed by both the enzymes, but one is the enzyme of urea cycle, two is the enzyme of pyrimidine synthesis. So the first reaction is this carbamoyl phosphate is synthesized, and in the further reactions of urea cycle, the urea non-toxic substance is formed from the toxic ammonia. The site is formed in the liver, only in the liver. There is a specific reason why only in the liver. I'll tell you during the urea cycle. Then it is, once it is formed, the urea, as it is non-toxic and more soluble, it is then released into blood and cleared by the kidneys, or filtered and excreted by the kidneys. Urea excretion depends on the protein intake. That means more the protein intake, more the excretion of urea, or vice versa. So protein intake affects the urea excretion in the urine. This is the first cycle discovered by the Krebs and Hensley. Even before TCA cycle, this is the first cycle, the urea cycle, found by these two scientists, Krebs and Hensley. So sometimes this cycle is also referred as Krebs and Hensley cycle. This urea cycle is a 5-step reaction cycle. First 2 steps occur in mitochondria and remaining 3 steps in the cytosol of liver cells. The 2 nitrogen atoms of urea are derived from, because the structure, if you talk about the structure of urea, if you talk about the structure of urea, it has one amino group, the carbon, oxygen, the keto group, and one more amino group. So this is the structure of urea. So there are 2 amino groups present, right? And one of the amino group is donated by the ammonia itself, okay? And the second amino group is donated by the amino acid aspartate. Okay, so 2 amino groups are present in the structure of urea. First is donated by the ammonia, which we are intended to neutralize, and second amino group is donated by the amino acid aspartate. Then urea is soluble, non-toxic, and it has high nitrogen content. So nitrogen from the body needs to be excreted, which is present already in the ammonia, which is toxic. So toxic is not converted to non-toxic, and less soluble to more soluble urea. So urea can be easily excreted from the body, and that's why it defines as a net excretion of nitrogen from the body. In the previous slide, if you remember, we have seen this term, net excretion. The genetic defects in the urea cycle enzymes leads to the condition called hyperammonemia, the genetic type of hyperammonemia we have seen. Newborns shows vomiting, ataxia and lethargy. If ammonia level is increased in the newborns, it shows these symptoms because in newborns the liver is yet to be matured. That's why hyperammonemia is seen or due to any genetic defect in the urea cycle. Significance of urea cycle: it is used for the detoxification of ammonia, toxic ammonia to non-toxic urea, biosynthesis of arginine from ornithine in liver, kidney and intestinal mucosa. Arginine is one of the 20 standard amino acids, right? So this arginine can be synthesized if required in the body from one of the metabolite of urea cycle, ornithine, in these three organs. Urea cycle is linked with TCA cycle. That's why sometimes urea cycle along with TCA cycle is termed as urea bicycle. Two cycles are connected to one another, sometimes referred urea bicycles, means amino acid catabolism is coupled with energy production, that is TCA cycle, through fumarate, because fumarate is formed in the urea cycle and it is utilized in the TCA cycle. So it is an example of integration of metabolism. Coming to urea cycle, as I told you, there are 5 reactions: 1, 2, 3, 4 and 5, catalyzed by the 5 enzymes, 1, 2, 3, 4 and 5 enzymes. So the initial product is ammonia and carbon dioxide, which through these 5 reactions finally it is converted to urea, and ornithine, which is utilized in the second reaction, is regenerated at the end of the 5th reaction. Its function is like oxaloacetate. So see, we'll go through one by one all the reactions. The ammonia is our starting point, and the carbon dioxide, it is the soluble form of carbon dioxide. So ammonia and carbon dioxide, by the action of the enzyme carbamyl phosphate synthetase, it is synthetase, that's why the ATP is utilized here. Because ATP is utilized, it is synthetase. So what is synthesized? Carbamyl phosphate. This molecule is a carbamyl phosphate, and ATP is converted to ADP. If you see, there are 2 ATPs utilized, converted to 2 ADP molecules. So the first reaction catalyzes formation of carbamyl phosphate from ammonia and carbon dioxide, catalyzed by carbamyl phosphate synthetase. This reaction happens in mitochondria. The second reaction is synthesis of citrulline. The carbamyl phosphate synthesized in the first reaction combines with ornithine. The ornithine is transported or transferred to this carbamyl phosphate, and combination product of these two is nothing but a citrulline. If you see the change in the structure, 1, 2, 3 carbons, 1, 2, 3 carbons and the amino group, and above this by liberating the phosphate, if you see the inorganic phosphate, the amino and keto group is attached. The carbamyl moiety is attached to ornithine, so it forms citrulline by the action of the enzyme ornithine transfer of carbamyl. To this ornithine, carbamyl is transferred, that's why ornithine transcarbamylase, the citrulline is formed. So this is the first reaction, this is the second reaction which occur in mitochondria, and the remaining steps occur in the cytosol. Now the citrulline, it is easily permeable to mitochondrial membrane through the protein transporter. The citrulline comes out of the mitochondria, and in the next step this citrulline is converted to arginosuccinate by the action of the enzyme arginosuccinate synthetase, and as it is synthetase, it requires ATP, which is converted to, remember this, AMP and pyrophosphate. This we already discussed during lipid metabolism, or I think is this, sorry, nucleotide metabolism. If ATP is converted to AMP and PI, it is equivalent to 2 ATPs. So citrulline is converted to arginosuccinate. Here there is addition of the full structure of the amino acid aspartate. As in the previous slide we have seen, the second amino group in the structure of urea is donated by this amino acid aspartate, which is added to citrulline, and citrulline plus aspartate becomes arginosuccinate. If you see the structure, this is citrulline. From this citrulline, at this position of the oxygen, the whole molecule of aspartate is added, and that is nothing but arginosuccinate, catalyzed by arginosuccinate synthetase. Now this arginosuccinate is lysed from this bond is broken. So from the aspartate, only this ammonia is retained in the structure of citrulline, this ammonia, and the remaining carbon skeleton is lysed, is removed, and that carbon skeleton is nothing but fumarate. Okay, and this fumarate, as we discussed, this can enter the TCA cycle, one of the metabolite in the TCA cycle, it is utilized there. And the enzyme that catalyzes breakdown of the arginosuccinate is arginosuccinase. It is lyase type of enzyme, and arginosuccinate, by liberating fumarate, itself get converted to the amino acid arginine. So difference between arginosuccinate and arginine is the absence of this carbon skeleton which is coming from the aspartate on the amino group present. So this is the arginine, and in the final step, again a lyase type of enzyme, arginase, breaks this arginine. You see these structure, this above structure, carbon and 2 ammonia. This ammonia came from this carbamyl phosphate or the initial ammonia, and the red color ammonia which is donated by aspartate in the subsequent reactions. So carbon and these 2 ammonia are lysed or separated. This bond is broken by the enzyme arginase. So this molecule is separated as urea, our final end, non-toxic end, soluble end product, and the remaining structure is nothing but the ornithine, which is utilized in the second reaction of the urea cycle. So it is regenerated. That's why its function is considered to be similar to that of oxaloacetate in the TCA cycle, which is utilized in the second reaction and again first reaction, and again it is regenerated at the end of the TCA cycle. As I told you, this urea cycle only happens or occur in liver. There is a specific reason, because there are 5 enzymes: carbamyl phosphate synthetase, ornithine transcarbamylase, arginosuccinate synthetase, arginosuccinase, and arginase. All the enzymes are present in all tissues except only one enzyme, which is present only in the liver, and that enzyme is arginase. That's why the urea cycle is completed, or urea is formed in only liver. Do remember this point, because it is important for the viva. Why urea cycle occur only in the liver? It is because the enzyme arginase, the last enzyme which catalyzes the formation of urea from arginine and regeneration of ornithine, it is present only in liver, not in any other organ. So this is the blank slide for the urea cycle. Please practice at yourself. Do read the urea cycle and practice by yourself answering the questions, asking the questions yourself and answering the questions. Then the fate of urea. What happens to urea? Now urea is formed from ammonia. What happens to it? Urea produced in liver is transported in the blood to kidneys for its excretion. The only function of urea in our body once it is formed is to get excreted. No any other function. Okay? And most of the urea is excreted in urine, but still some amount reaches to the intestine, and in the intestine, by the bacterial enzyme, action of the bacterial enzymes, this urea is converted to ammonia. Okay, the enzyme urease present in the bacteria. So one of the source of ammonia that we discussed. So in renal failure, urea is not properly excreted, leading to uremia, because the kidney, the important function of the kidney, along with the excretion of excess water, it excretes the unwanted waste substances produced in the body. And one of the important or major waste substance produced is urea. If there is any defect in the functioning of kidney, say for example filtration, GFR is affected, urea is not effectively filtered and excreted. Instead, it is retained in the blood, leading to increased level of urea, which is called as uremia. And in intestine, large amount of urea is broken down to form ammonia, which is absorbed in the blood, leading to hyperammonemia. So this reaction shifts towards the right hand side and ammonia is formed, and this ammonia absorbed in the blood, leading to increased level of ammonia, that is hyperammonemia. Thus in renal failure, as urea is not excreted in the urine, rather it takes the other path, that is intestine, catabolized to ammonia, ammonia comes to blood, causes hyperammonemia. The urea normal range is 15 to 40 milligram per 100 ml of serum or plasma. The condition in which urea level is increased than the normal, that is above 40 milligram per deciliter, the condition is called as uremia, not hyperurea. Remember, for urea increased level, the condition is named as uremia. Uremia can be of three types, because the urea is formed from proteins. It is the end product of protein metabolism. We take it through diet. So till it reaches the kidney, that is the formation of urea. Once urea is formed, the kidney's central function is to filter it and remove from the body in the form of urine. So central organ is kidney for its removal, and once it is filtered through kidney, it is excreted through ureters to urinary bladder through urethra in the form of urine. So there are three stages of urea formation, filtration and excretion. So depending on where the defect lies to cause uremia, depending on this, there are three types of uremia. Keeping in mind the central organ, that is kidney, the names are given by the term pertaining to kidney, that is renal. If that means central organ is renal, if the urea level is increased due to some reason which comes before kidney, that is excessive production of urea from proteins, it is called pre-renal uremia. It may happen due to decrease in blood volume. The urea level may be normal, but due to dehydration or due to congestive heart failure, coronary heart diseases, the blood volume is decreased. In this case, there is relative increase of urea, not absolute increase. Because blood volume is decreased, urea level is normal, but blood volume is decreased, so urea seems to be increased, but actually it is not increased. Right? It may be 15 to 40 only, but whatever the denominator per 100 ml of serum, suppose 5 liter of blood is present in a given person, 5 liter of blood present, and in that case the urea level is suppose say 30 milligram per deciliter, but the urea production remains same 30 milligram only, but if the blood volume say decreases to 3 liters, so as compared to 5 liter, in 3 liters of blood, urea 30 milligram, the concentration won't be 30 milligram per 100, it will be say in comparison to volume, but it is 30 milligram actually. So it is relative increase. It seems the urea level is increased. Increased tissue protein breakdown, as in the illness, GI bleed, in the diabetic patients, protein breakdown increases, and the end product of protein is urea. When we take high protein diet, more protein, more conversion to amino acids, more conversion to amino acids, more formation of ammonia and more formation of urea. But it is a physiological condition. Once all the protein is digested and converted to urea, once excess of urea is excreted, the urea level is maintained to normal. The second type of uremia is renal uremia. Here the defect lies in the functioning of kidney, in the kidney itself. So due to improper functioning of kidney, the conditions like kidney failure, it may be acute chronic glomerulonephritis, nephrotic syndrome, renal tuberculosis, renal cancers. So all kidney diseases, the urea level is increased because it is not effectively filtered and excreted by the kidney. And the last is post-renal uremia. Obstruction in the pathway of excretion causes urine retention and reduces effective filtration pressure. So once urea is filtered through the kidneys, it reaches ureters, bladder and urethra. So any blockage in any of these, like stones, kidney stones, ureter stones, stones in the urethra, that blocks the urine passage, leading to no excretion of urea, and by backflow or the urea again in the kidneys not effectively filtered, rather it is accumulated in the blood leading to uremia. Various reasons for the obstruction of the urinary passage include enlarged prostate because it is situated near to the excretory system. Enlargement of prostate may block the ureters. Stones in the urinary tract anywhere, mostly ureter or the opening of the ureters in the bladder or in the urethra. The stricture of urethra, due to in aged people the opening of the bladder or the last passage of the urethra is blocked or compressed. It may be due to fistula. So decreased blood urea level, one thing we have seen is uremia, increased blood urea level, so decrease than the 15. It is due to low protein diet. If you take low protein diet, low amount of protein is converted to low amount of urea. Also liver diseases because urea is formed in the liver, only in the liver, and if liver cells are damaged, the conversion of ammonia to urea is affected. So in liver diseases also decreased amount of urea is seen, and apart from these two, the decreased level of urea is clinically irrelevant. So to summarize ammonia metabolism, the sources are bacterial action in the intestine, trans-deamination, the purines and pyrimidines once catabolized also adds to the ammonia. The other amines and other nitrogen containing compounds also adds to ammonia. The next is the fate of ammonia. What happens once ammonia is formed? The first is and most important is the formation of urea and formation of glutamine, synthesis of purines and pyrimidines, excretion in urine as ammonium salts that gives the characteristic smell to the urine. Then the transport, there are two major transport forms. One is glutamine in the tissues and the brain majorly, and second is alanine in the muscles to liver. So this forms the glucose alanine cycle. So to summarize, till now the proteins by the action of the enzyme proteolytic enzymes converted to amino acids in the stomach, in the intestine by various these enlisted enzymes along with hydrochloric acid. So these amino acids undergo transamination, right? Transamination is nothing but the transfer of amino group. There is no removal of ammonia. So transamination followed by deamination. In this step, the ammonia is liberated, and this glutamate, glutamine, and alanine, these are the they have the ammonia, which in the further step all are converted to glutamate or adds the ammonia by transdeamination or deamination, and in the liver itself the ammonia is converted to urea by urea cycle, and this urea, its fate is to get excreted in the urine via kidneys. And once these amino acids undergo transamination, the amino group is transferred, but the carbon skeleton of that amino acid is converted to alpha keto acid. That alpha keto acid may be utilized for the synthesis of ketone bodies or fat if it is a ketogenic amino acid, or it is converted to glucose or the carbohydrate if it is a glucogenic amino acid via gluconeogenesis. The alpha keto acids like pyruvate, oxaloacetate, alpha ketoglutarate, all these are converted to glucose by gluconeogenesis. So this is the summary of till now what we have covered: the digestion, absorption, the common reactions of the protein metabolism, including transamination, deamination, and urea cycle. I hope